Lightning is one form of energy. It's not one we can harness to do cellular work, though. Batteries, wind turbines. The center panel shows different forms of energy conversion, changing energy from one form to another. This is the last chapter before exam 2. Remember, membranes aren't just dividing the inside from the outside. They are places where many cellular functions happen. But they are also dividers. Everything that comes into a cell or leaves the cell must pass through that membrane. Membranes are predominantly phospholipid bilayers. The arrows are pointing to the heads of those molecules facing inward toward water and outward toward water, protecting the hydrophobic tails. We mentioned phospholipids back when we were discussing macromolecules. Lipids are one of your 4 major macromolecules. Triglycerides had 3 fatty acids attached to a glycerol. Phospholipids have 2 fatty acids, and the glycerol, and a phosphate group. The phosphate group, as usual, has a negative charge to it. This makes the head hydrophilic. The tails of the fatty acids are nonpolar hydrocarbon chains. This makes them hydrophobic. Since most cells are about 2/3 water, this is a problem for the hydrophobic ends. So what happens is that the molecules arrange themselves in a double layer, tails together and protected by the water within and without the cell, and heads out toward the water. This arrangement causes the membrane to be selectively permeable. That means that some types of molecules can pass through the membrane fairly easily, others can't. The phospholipid bilayer and other components of a membrane compose a fluid mosaic model. Fluid because all the molecules are in motion. A mosaic because of the many small pieces that compose the membrane. Visualize, if you will, all those plastic balls over in the McDonald's Playhouse, all different colors. Call the yellow ones phospholipids. Call the red ones proteins. The green ones, cholesterol. Orange, glycolipids. Now, visualize taking all those balls and tossing them in a swimming pool. Fill the surface of the water with balls. They're all floating on top of the water. The wind is blowing. Although the number of phospholipids will not change, and neither will all the rest of the components, they don't stay in the same place either. They move, they bump into each other, they shift places. This is like one side of a bilayer. Now, imagine a second layer with the same composition floating under the first. And add some bigger balls that will show up on both sides of your bilayer. The wind is moving both layers around. Sometimes a molecule will even switch layers. Now you can see why the membrane is called a fluid mosaic. Lots of things happening here. You can see phospholipids, embedded cholesterol, which is only found in animal cells, and embedded proteins. Note that the different proteins illustrated have different jobs. Some are assisting in holding cells together. Some are transporting molecules from one side of the membrane to the other, either passively or actively. We'll look at the difference in those shortly. Some are enzymes. Others are transferring signals. Remember that one of the functions of proteins is signaling. Something has to receive those signals, and here it is, another protein. The signaling molecule locks onto the outside of the embedded protein, and the message is transferred inside to make things happen. What things? Remember puberty? Those raging hormones were signaling molecules. They went all over your body, causing physical, emotional, and mental changes. Also, notice the cytoskeletal fibers just under the membrane? Those assist in cell movement. Those proteins that go all the way through the membrane, they have jobs. One type are enzymes embedded in the membrane. Remember, smooth endoplasmic reticulum has embedded enzymes to help synthesize lipids and detoxify. Some are for signal transduction, the sending of messages. Molecules outside the membrane that fit a receptor on a protein may trigger a reaction inside the cell or inside the organelle. This is a response to the environment. Another type is for transport, and there are a couple types for that alone. We'll look at those in a minute. Just another view of membranes. For diffusion, we often use the example of a smell. If I opened a bottle of perfume in your room, it might take a minute, but you'd be able to smell it across the room in a bit. The molecules in the bottle are concentrated in one place. They are, like most molecules, in random motion. That random motion and bumping into each other will send the molecules out to where there are fewer of them, moving the molecules from an area of high concentration to an area of low concentration. This is a concentration gradient. Molecules naturally move down the concentration gradient, high to low. It takes no energy to have this happen. It's a natural occurrence of the natural random motion of molecules. If it takes no energy, it's considered passive. The same principle applies here with some variations. The molecules that move in living organisms aren't just moving through the air. They're moving across membranes. More comes into play with this. Visualize opening a box of tennis balls at the top of a hill. Random motion, no energy, area of high concentration to low. The vertical gray dashes are representing a semi-permeable or selectively permeable membrane in fluid. Remember, that means some things can go through while others cannot. On one side of the membrane, green dye has been placed. It is a molecule that can go through the membrane, so as the molecules move, some go through. Notice that some go back. This is random motion, so although the net movement is down the gradient, molecules are going all over. Eventually, however, equilibrium will be reached with approximately the same number of dye particles on either side of the membrane. The molecules are still in motion. Now we've placed different colors of dye on either side of the membrane. Think back to the perfume example. What if I opened a bottle of perfume on one side of the room and a bottle of skunk scent on the other? Will they stop when they meet each other? No, you'll have a horrific blend of scents in that case. But the same holds true here. The two molecule types basically ignore each other. The green dye will reach equilibrium with the green dye. The purple dye will reach equilibrium with the purple dye. They do not affect each other. The diffusion of water has its own name, osmosis. When the solute, the molecule, cannot cross the membrane, but the solvent, water, can, the solvent moves instead of the solute. Look at the diagram. In this case, the molecule or solute in question cannot go through the selectively permeable membrane because it's too big. The bottom diagram shows what the molecules are doing. The polar water molecules form weak hydrogen bonds with the solute molecules. On the side with more solute, the water molecules are busy. They're clustered around the solute molecules. On the side with less solute, there are more free water molecules that can travel across the membrane. Some more terms to know. Tonicity also refers to the number of solute particles, which influences a cell's tendency to gain or lose water. We speak of cells in a solution. This solution is the extracellular matrix for many. It may be water itself for a free-living cell. The cell is bathed in a solution of some kind to keep it hydrated. Break the words down. Tonic, number of particles. Iso, same. Hypo, fewer. Hyper, more. Solution, a solute dissolved in a solvent. So a hypertonic solution has more particles in the solution. A hypotonic solution has fewer particles in the solution. And an isotonic solution has about the same number of particles in the solution. More, fewer, and same are all words of comparison. What are we comparing this solution to? The cells within it. This has animal cells on top and plant cells on the bottom. In a hypotonic solution, water moves into the cell through the membrane in an attempt to reach equilibrium. If too much moves in, an animal cell might lyse. It can only stretch so far before it breaks. Remember, that's the root word of hydrolysis, to break. A plant cell has a wall to support it, but it will stretch until it is full and tight or turgid. In a hypotonic solution, water moves from the solution into the cell. The net gain is into the cell. In a hypertonic solution, this animal cell will dehydrate, losing water to the solution outside. The plant cell will as well, but since the membrane is attached to the wall, it also undergoes plasmolysis, the pulling away of the membrane from the wall. In a hypertonic solution, water moves from the cell into the solution. The net gain is to the solution. In an isotonic solution, equilibrium is already there, but the molecules are still in motion. There is no net gain in either direction. Do you know how if you're eating ice cream, you get thirsty? The sugar causes a hypertonic solution in your mouth, so the cells in your mouth are dehydrating. That makes you thirsty. I know everyone has heard that you shouldn't drink salt water. If you're in a lifeboat after your cruise ship goes down, you're better off not drinking anything at all. Drinking the salt water puts a lot of hypertonic solution into your system, so your cells are losing water trying to reach equilibrium with it. You dehydrate much faster. This time, you're in a hypertonic solution. The water moves from the cell into the solution. The cell shrinks. I have added additional osmosis problems on Canvas under Files. If you need more help, let me know. Some things would move down the concentration gradient if they could, but they may not be able to cross a membrane. For those molecules that are needed inside the membrane, there are proteins embedded in the membrane that form channels, which allow the needed molecules to enter by passive transport down a concentration gradient. These molecules are essentially holding open a door, and that takes no energy at all. It's still passive transport, just facilitated, helped by the protein. Any molecule that can fit can go through these open channels in either direction, in or out. All of the movements of molecules we've looked at so far were passive transport. No energy required moving down a concentration gradient, facilitated or not, including the osmosis of water. Now let's look at how molecules move up or against a concentration gradient. This one takes energy. The cell is doing work, moving against a force. The force here is gravity. I point out to my face-to-face students that if I wanted to pack all of them into the closet down the hall, that would take energy on my part. If I opened the door, I would not need to expend energy, as they would come out and spread out on their own. The difference between using energy, active, and not using energy, passive. Active transport takes another embedded protein, a transport protein. This protein will only allow solutes of certain shape to enter it and bind. So each molecule required in the cell has its own transport proteins, since shape determines function. The protein for molecule A will only work with A. If we want molecule B brought in, it needs its own transport protein. The correct solute binds to the protein. ATP, the main energy molecule, transfers a phosphate group to the transport protein. This releases energy, which is used by the protein to change shape. Changing shape alters which way the protein is opened, and the molecule can exit into the area of higher concentration. The protein then resets itself, releasing the bound phosphate group, which is then free to go be recombined with an ADP to create another ATP molecule. Since ATP energy is used, this is active transport. This is another way items can be moved through the membrane for particles too large to fit through a transport protein. Exocytosis is the removal of large materials. It may be proteins being excreted, such as insulin. It may be materials for the extracellular matrix being excreted, or waste. Lots of possibilities. A transport vesicle containing the material to be excreted merges with the plasma membrane so that there are no leaks in the cell and the molecule is released outside the cell. X for exit, cyto is cell, exit the cell. A pocket forms on the plasma membrane, trapping molecules from the solution on the exterior. This is called an invagination. It's not very selective of what molecules get trapped. It builds inward, then closes off to form a transport vesicle. The molecules are then taken to the appropriate organelle. N for in, but don't misspell it. There are specific types of endocytosis. Phagocytosis is specific to bringing in food particles. Pinocytosis is specific to bringing in water. Receptor-mediated endocytosis uses receptor proteins in the membrane. They capture specific molecules, collect together on the surface. Remember, all the membrane molecules are moving, fluid mosaic. Then the invagination brings the cluster inside the cell. Here are some YouTube videos that might help. Go surfing. Many will be more detailed than this, though, so don't dive too deep unless you can swim. For our body, the opposing force might be gravity or friction. Just picking up your water bottle is work. You're moving it against gravity. You aren't rearranging matter in this case, but that is what happens in chemical reactions, the rearrangement of matter. Kinetic energy is the first of two forms of energy. There are also two types of kinetic energy, heat and light. Heat is one form of kinetic energy associated with the random movement of molecules or atoms in a body of matter. While you can warm your body with this heat, cells can't use it as an energy source. Light is another type of kinetic energy, and this one can be harnessed by living organisms. The second type of energy is potential energy. This is stored energy. This could be a ball at the top of a hill, or the electrons on the outer orbit of a molecule. Chemical energy, or the potential energy of molecules, is based on structure for the most part, although we will see in later chapters how it can also be from location. A cell is just a tiny chemical factory. Some chemical reactions assemble monomers into polymers. Some chemical reactions break sugars and other fuels to release energy. Cells are energy transformers, converting one type of energy into another. Exactly. If energy could not be converted, we could not